this video, we'll take a look at proteins. Proteins are polymers that are composed of building blocks called amino acids. So the monomer of a protein is an amino acid. Proteins have a diverse range of functions. They can be regulatory in function, structural, protective. They can function in transport. They can function as enzymes and toxins. First, we'll take a look at protein function and enzymes. Enzymes are these proteins that catalyze biochemical reactions, which means they speed them up. So they affect the rate of the chemical reaction, speeding it up so that that reaction can occur in such a way that will support living organisms. These reactions need to occur at a pace that will support life, and these enzymes allow those reactions to occur at that rapid rate. Enzymes are very specific. The molecule that the enzyme works on is called the substrate, and they will be very specific for their substrate. We have two major classes we can think of. We have catabolic enzymes. These are enzymes that break down substances. So think about this in terms of digestion. We also have anabolic enzymes. These are enzymes that build more complex molecules. Think of building muscle tissue. That's anabolic. So catabolic is breaking down, anabolic is building up. This can be a little confusing when you first look at the prefixes here because we tend to think of cat as positive and an as negative. The way to think about this, if you want to try to make sense out of it, is that these names were first given when they were studying digestion. So from the perspective of digestion, breaking things down is kind of going in that positive direction. And so they referred to these as catabolic. And then when we build more complex molecules in terms of if your focus is digestion, that's going the opposite direction. So they were given the term anabolic. Here we see a picture of an enzyme. This is an enzyme that catalyzes the conversion of maltose to glucose. So it breaks maltose, which is a disaccharide, into its component glucose monomers. This table gives us some protein types and their functions. We have digestive enzymes that break down molecules into their monomers. Lipases work on lipids. We also have transport proteins. A good example of a transport protein is hemoglobin that carries oxygen throughout your bloodstream. We have structural proteins, which are there more for physical structure. Collagen, keratin. These form physical structures in the body, like tendons and ligaments and hair. Then we also have hormones. Now, not all hormones are proteins, but we do have some hormones that are, like insulin and thyroxine. Hormones typically coordinate the different body systems with one another so that the different body systems work together as a whole. We also have defense proteins like immunoglobulins. So when you're exposed to a foreign pathogen, your body produces an antibody, which is an immunoglobulin, which provides defense. We also have contractile proteins that we find in all cells for cell division, and then we find very specific arrangements of these contractile proteins in cells of muscle tissue. We also have proteins for storage. Many of these will provide nourishment to developing embryos and developing seedlings. Let's take a look at amino acids. Amino acids are the monomers that make up these proteins. Like we had before with carbohydrates, the monomers were monosaccharides. Here, the monomer is an amino acid. This is the fundamental structure of an amino acid. There will be a central carbon, often called the alpha carbon. There will be an amino group over here. This NH2 is the amino group. There will be a carboxyl group. This is the carboxyl group, COOH. You'll also have a hydrogen on that central carbon. And then you'll have an R group or a side chain. This is a variable portion, so all of this other part will be the same for all amino acids. What will differ from one amino acid to the other is what sits here on this side chain. These amino acids have diverse chemical properties depending on which amino acid we have. There are 20 different amino acids that we commonly see in proteins making up protein structure. This shows those 20 different amino acids. Each amino acid has that same basic structure we looked at before. It's got the central carbon, the amino group, the carboxyl group, a hydrogen, and then in blue here we have that variable portion, which is the R group. So what makes one amino acid different from another is what's sitting in the R position. And it's those R positions that will determine the behavior of that specific amino acid. The other part of that amino acid will behave very similarly from one amino acid to another. But the R group will behave quite differently from one amino acid to another. Now, some amino acids might behave somewhat similar because they have somewhat similar R groups. Notice we have groupings of the R groups here. We have some R groups that are nonpolar aliphatic R groups. Remember, aliphatic means it's a chain rather than a ring. They're nonpolar, so it's essentially a chain of hydrocarbons. These would tend to behave more like one another than they would, say, one of these polar ones. We also have nonpolar aromatics. Remember, aromatic means that it forms a ring. They're still nonpolar. They're still going to behave the same way in water, but they are ring structures instead of chain structures. Then we have some that are polar. So here we have polar R groups. That means that they're going to have these partial charges. That means they can interact with other groups that maybe are also polar and have these partial charges, or with other ions. Then we have some that are ionic and positively charged up here. And then we have others that are ionic and negatively charged. So each one of these will have unique characteristics based on that R group. These amino acids are typically represented in one of two ways, either by an uppercase letter or by a three-letter symbol. So valine can be symbolized as a capital V or VAL. Another example, aspartic acid, can be symbolized as a D or ASP. When you take biochemistry, you will become very familiar with these three-letter designations. They will probably become as familiar to you as Avogadro's number is right now, or the chemical symbol for carbon. Some amino acids are essential. Remember, we talked before about essential fatty acids, meaning that you had to have them in your diet. The body needed them. You need them in the diet because you can't synthesize them. Same thing for amino acids. These are amino acids that we need. We cannot synthesize. They have to be in the diet. For humans, the three essential amino acids are isoleucine, leucine, and cysteine. Other animals will have different essential amino acids. Now, the sequence of amino acids in a protein and how many amino acids are in that protein will determine the overall shape of the protein, the size, and its function. Let's take a look at how we join those amino acids together. When we hook these monomers of amino acids together, we create a bond called a peptide bond. Remember when we hooked together carbohydrates, we called it a glycosidic linkage. When we hooked a fatty acid onto a glycerol, we called it an ester linkage. So we had glycosidic linkages in carbohydrates, we had ester linkages in lipids, now we have peptide bonds in proteins. Let's take a look at the peptide bond formation. Up at the top we have two amino acids. We're going to join those, and the way we're going to join them is we're going to take the carboxyl group of one amino acid and the amino group from its neighboring amino acid. We're going to remove water, so once again it's a dehydration synthesis, so you should be seeing a pretty strong pattern here. A dehydration synthesis, remove that water, and create the bond here between this carbon and that nitrogen. And that bond is that peptide bond. When we do this, of course, we're going to release this molecule of water. So we start off with two amino acids. We end up with a dipeptide because we have two of them down here joined together by a peptide bond and a molecule of water. To make a polypeptide, we would just continue to do this over and over and over again, linking together many amino acids. Now, what is the difference between a polypeptide and a protein? In some cases, they can be the same thing. In other cases, they're not. A peptide is a chain of these amino acids. So if I take that dipeptide and then I just keep adding another amino acid and another amino acid and make it many, many, many of those, I create a polypeptide. A protein can be that polypeptide, or it can have several chains. So I could stop at just this polypeptide chain, there's my protein, or I could create several polypeptide chains and hook them together to create a protein. So a protein is one or more polypeptide chains. Now sometimes, even if you only have one polypeptide chain, you might modify that chain a wee bit. So you might go and add some other, what they call prosthetic groups to the chain. They might be non-peptide prosthetic groups. For instance, hemoglobin has a prosthetic group of iron in it. Each protein will have a unique structure, a unique shape that will dictate its function. Let's take a look at protein shape. Now the shape is crucial to the function of a protein. If you change its shape, you are likely to make it so that it is no longer capable of functioning. The shape is based on 4 different levels of structure. We call these the primary structure, secondary structure, tertiary structure, and quaternary structure. So we've got first, second, third, and fourth levels here. Let's take a look at the primary protein structure. The primary structure is the sequence of amino acids. So if you write out the sequence of those amino acids, you're telling me the primary structure of the protein. So we can see a picture of it here. Imagine a chain of beads, and each bead represents an amino acid. So we might be making this long chain of beads that consists of 20 different colored beads, each color representing a different amino acid. This example is bovine serum insulin. The sequence of the amino acids is unique to this protein. If we were to alter this sequence of amino acids, it could alter the function of this protein. So if we don't have the same order of our amino acids, even if we had all the same ones there, if they were in a different order, it could drastically change this function. Now, how do we get that primary structure? What determines the primary structure of the protein? It goes back to genetics. So back in DNA, we have genes, sections of our DNA, that have instructions for how to make each of these different proteins. So we say that a gene codes for a particular protein. If I change my DNA, so if I have a mutation in DNA, so that the sequence of DNA is now different, the gene is different, it could give me a different amino acid sequence. And if it gives me a different amino acid sequence, it could mean that this protein would no longer have the same structure and would possibly no longer have the same function. So when we see mutations in DNA resulting in physical changes, this is the mechanism by which that happens. Let's take a look at an example of that, sickle cell anemia. We have a change in one amino acid in this protein that has a very, very profound impact on human health. This looks at just a small part of the hemoglobin chain. In normal hemoglobin, at position 7, you have glutamic acid. In an individual that has sickle cell anemia, this glutamic acid is replaced by valine. So everywhere else in the chain is identical between an individual that has normal hemoglobin and one that has the sickle cell hemoglobin. There's one change in an amino acid along that chain, and that has a profound effect. We have 147 amino acids in this beta chain of hemoglobin, yet that one change leads to sickle cell anemia. When we look at sickle cell anemia, what we find is that the cells are shaped differently. You can see a good example of it right here. You can see slight crescent shape over here. These red blood cells that contain the sickle cell hemoglobin form this crescent shape, this sickling. It means that they don't carry oxygen as well. It also means that this shape causes them sometimes to get stuck in small vessels, and there can be some very severe health consequences for both of those things, lack of oxygen delivery and then also clotting. Let's take a look at the secondary structure of a protein. The secondary structure has to do with coiling or pleating of our polypeptide chain. This will be the result of hydrogen bonds that occur between the carboxyl group of one amino acid and the amino group of another amino acid. If we look at this chain, this is an amino acid. There's its amino group. Here's a different amino acid, its carboxyl group. This dotted line represents a hydrogen bond between those. Down here, the same thing. We have a polypeptide chain across the top. We have another polypeptide chain below it. And there's an amino acid here whose amino group is hydrogen bonding to another amino acid down here's carboxyl group. Remember, these amine groups and the carboxyl groups are the non-variable portion of an amino acid. So these are interactions that really don't depend on what R groups are present. There's two different types of secondary structure that we see here that tend to form. We have the alpha helix over here. The way the alpha helix works is that these hydrogen bonds that we see forming between these amino acids occur along the length of the chain between about every fourth up position along the chain. We have a chain of beads. If I grab the first bead and then I go 4 beads down and I take that bead and I allow it to come back and almost like a magnet hook on to my first bead. And then I go down another 4 and I hook it back around. And so it causes it to coil, almost like if you were imagining coiling up a garden hose. You're holding it in your hand, you form a loop, you bring it back around and you grab it. And then you make another loop and you grab that. So that's what that coiling looks like. And that's that alpha helix. The other type is a beta pleated sheet. That's what we see down here on the bottom. Now what we have is a polypeptide strand up here, another one below it, but now they are forming hydrogen bonds between these strands. And what it ends up doing is causing it to end up looking sort of like a folded paper fan. That's what we mean when we say a beta pleated sheet. It kind of looks a little bit like folding up a piece of paper in a sort of a zigzag. Here, the hydrogen bonds are between sections of these chains that are forming sort of these cross links here, holding them together into that sheet-like structure. Whether or not you get an alpha helix or a beta pleated sheet will depend on the amino acids that are present. Some amino acids are more likely to form alpha helices. Other amino acids favor the formation of the beta pleated sheet. Next, we have the tertiary structure. This is the third level. In our tertiary structure, we start to create this three-dimensional structure of the polypeptide. Imagine what we have is this beaded chain, and we've created an alpha helix. So we now have a beaded chain that is a spiral. Now we're going to take this beaded chain that's a spiral, kind of like a slinky with beads on it, and we're going to fold it up. So we're going to just fold different sections back on each other. That's what our tertiary structure will look like. Inside of here, we have this coiled chain, and then we're going to fold pieces of that chain back on itself. Now, this is going to be primarily due to those R groups that are the variable portion of the amino acids. Our secondary structure had to do with interactions between the non-variable portion of an amino acid, the carboxyl and the amino group. Now we're looking at interactions between those side chains that are coming off, those R groups. And because they're so variable, that means that this tertiary structure can be highly variable. So remember, we only had 2 basic types of secondary structure, the helix and the beta pleated sheet. Tertiary structure, it's all over the place because there's so many different types of R groups. There's so many different ways they can interact. So let's take a look at what creates this tertiary structure. There's a variety of different types of chemical interactions that do this. One is hydrophobic interactions. Remember, some of our R groups were hydrophobic. Those hydrophobic R groups are going to tend to cluster together on the interior of the protein. A lot of the rest of the protein is polar or charged. And so these nonpolar groups tend to go away and form these little interior pockets. We have these hydrophobic R groups here forming this interaction with one another. We also, remember, had some R groups that were charged. We had positively charged, we had negatively charged. These can form these ionic bonds if we have opposite charges. So if I have a positively charged R group near a negatively charged R group, I could form an ionic bond here. Likewise, if I had 2 positively charged areas, they would repel one another. We also can get hydrogen bonding. Just like we could get hydrogen bonding between the carboxyl and amino groups, we can also get hydrogen bonding between R groups, depending on what R groups are present. And that would tend to cause this to fold back on itself. The other type of interaction we can get that creates this tertiary structure are called disulfide bridges. Cysteine is an amino acid that has sulfur in it, and sulfur, like oxygen, likes to form bonds with itself. And so if I have a cysteine sitting here and another cysteine sitting there that has a sulfur group off of it, they will often tend to form these disulfide bridges that will again cause folding of that molecule and cause them to stick together. Now let's take a look at that fourth level of protein structure, the quaternary structure. This is going to be the interaction of several polypeptide chains put together. Imagine that each one of these, the pink here, this one pink, and then there's another pink one up here, each one of those represents one of those beads that are in a coil that are then folded up. So it's got a primary, a secondary, and a tertiary structure all in this one pink chain here. It's got another primary, secondary, and tertiary structure all in this pink chain over here. Same thing for these two blue chains. So I've got 4 chains that are folded there. Then I can have those chains stick to one another and interact to create this fourth level. These typically are fairly weak interactions here. Not all proteins will have quaternary structure. Some proteins will just have a single polypeptide chain. But if they do have multiple polypeptide chains, then they will have a quaternary structure. It depends whether or not they have one polypeptide chain or more than one polypeptide chain. Let's review these 4 levels of protein structure. We had our primary structure, we see here. That was that linear sequence of amino acids, sort of like a beaded chain. Then we can take that and we can either twist it into an alpha helix or we can fold it into a beta sheet. This only shows us that helical structure, but this is that secondary structure. And remember, that's due to hydrogen bonding between the non-variable portions of our amino acids, the carboxyl groups and the amino groups of 2 different amino acids on the chain. So now I've taken my beaded necklace and I've twisted it, or I've coiled it into a helix. Now I'm going to take that coiled chain and I'm going to create the tertiary structure by folding that twisted or pleated chain. I'm going to fold it now back on itself. Then I'm going to create the quaternary structure by taking several of these coiled chains and hooking them together. So we can say this is a combination of several folded, twisted chains. What I've done here with our definitions is I've tried to make each definition incorporate the one prior to that. So here we had a linear sequence of amino acids. Here we twisted or pleated that linear chain. Here in the tertiary, we folded the twisted or pleated linear chain. And then in quaternary, we had a combination of several of the folded, twisted, or pleated linear chain. Each one incorporates the level below it in terms of this sequence. Let's talk a little bit about protein folding and denaturing. If we go in and break some of these chemical interactions, those ionic interactions, those disulfide bridges, hydrogen bonds, what will happen is the structure of that protein will begin to change. Its shape will change. There's several factors that can cause these changes in shape. Changes in pH, changes in temperature. Either one of these can cause what we refer to as denaturing of a protein. In denaturing, the protein changes to a point where it no longer has the proper function. Consider it denatured if it doesn't function anymore. In some cases, that denaturing might be irreversible. So we cannot uncook an egg. When you apply heat to an egg, the changes that you're seeing there to the egg white, that is a denaturing of that protein. So that is an irreversible denaturing. So that gives us a brief overview of proteins.